Good afternoon, everybody. So this is a breakout session for the Asian HCA. Okay, so I introduce myself. I'm Chung-Chau. Me, myself, is from Hong Kong, but now I'm working in Japan, in Weekend, with Jay and Piero on organizing the humans at Alice. So me, myself, is a bioinformatician, specialized in looking into this non-coding element as well as human genome evolution. So today's breakout session here is about bioinformatics and databases. So I think we could discuss something that is not necessarily has to be only applicable to this Asian community, but ourselves as part of the global HCA community, that how we could work together with maybe the DCP, or maybe contributing some ideas, how we could come up with this Human Atlas 1.0. So the discussion is pretty much free. So how it works here is we would have I would bring up several questions that probably some of you might have some opinions, and then you could pass the mic and say about your opinions, and then Xuegong over there would drop down whatever that you guys are raising up, and then we will do a summary at the end and combine with the other breakout session. Okay, so we could start with something more general. Okay, so in terms of like data standardization, cell annotations, as well as data deposition. So some of us already started some international consortium between different groups, say for example IDA. Okay, so that we were thinking about whether there is like should be a standard way of annotating the cells, for example. Okay, so one thing that you guys might be worth bringing up about is say cell annotations. Okay, in some of these local consortiums, is there anything that's needed to be standardized, for example? Okay, so, and the second thing is also about, we've been discussing among ourselves, some of the work that we've been doing is part of the HCA, but we ourselves also want to share data among ourselves. Okay, so, but could we, does it necessarily has to be deposited into the DCP, or we should come up with some of the our own data deposition for our own consortium, for example, the Japanese HCA. So this is something that, if you guys have anything you want to talk about, want to say about it, that we could probably bring it up here. Or any other topics. Not necessarily has to be related to the Asian HCA. Yeah, go ahead.
So I may have missed this, but the DCP will be a global DCP, or are we having a different DCP for Asia? Because if there are disease involved, then...
Right, so a good question. So we take what we've done with the Japanese medical single-cell network, for example. So the problem with the DCP is, for now, everything is deposited, it's 100% open. I'm sure that they're also working on the controls access. Okay, so, and now the DCP in general, I'm referring to the global DCP. Okay, so, but I also noticed some of you guys, like the Great Bay Area, they have their own network. Okay, so that whether the data generated that has to be deposited into the DCP, contributing to the global HCA, or some of the consortium would be more preferred to have a controlled access of their own data before, they publish their data. Okay, so this is up for discussion. Okay, so and I think also there's one other possibility is if this local consortium has enough communication with the DCP that we might be able to like integrate some more customized control access to that particular consortium's data. Okay, so this is something where to be discussed, I think.
Hi, yeah. So there's a few DCP people here in the room. I'm one of them. So I'm going to ask the DCP people to wave hi. There we go.
Right there.
So
There
there's
you go.
Tim and Kylie down here, then
There you
Saman,
go.
Paris, Will, and Clay at the back, and I'm Tony. So we're covering kind of like the Broad and UCSC and EBI here in the room. So I think we'd be really interested to hear from people about actually using the DCP for sharing data and for as a collaboration tool. So that is something that I think we'd be really keen to hear about, if there's kind of things that people need or challenges that people have with actually sharing the data and using the data. I've heard the sort of open access, controlled access as being one of those things. This would actually be a useful time to sort of talk about that. So there's plenty of people in the room here who can answer those questions if you've got them. So I will open the floor up if people want to get into that.
I was just thinking, has anyone here in the audience actually used the DCP? I've got one. Maybe, so we've got one qualified user to give you comments.
So do you have plans to make some data accessible only through some kind of a security, some sort of a private data set?
Yeah, hi. Jonas sort of touched on this morning. So this is a thing like the sort of controlled access, managed access permissions for data in the DCP is something that is an ongoing conversation. I don't think I'd want to say too much more about that. I think we're aware of the need that people have, I think it is a challenge. I think it's a challenge we're trying to work our way through.
So if that's the case, I don't see that much advantage in having a separate data deposition for Asia. Well, I mean, you'll duplicate a lot of the efforts. I just don't see what the advantage would be.
I think it depends on how long that conversation is going to go for. If it's going to be finished in a month, then that's great. If it's going to be finished in 3 years' time, it's too late.
Yeah, I mean, I think the big challenge is kind of the sort of alignment of the various like regional regulations. So, what's relevant. I mean, I come from the European Bioinformatics Institute, so much more familiar with GDPR, whereas we have other organizations that are familiar with FISMA in the US, and there is a challenge around aligning this. And there's also a challenge of as Jonah talked about in his presentation this morning, making sure that we've got very open access to all of the data that's available, and that's kind of somewhat at odds with the kind of restrictions over sort of private data sharing. Obviously not. There's sort of two different issues there, right, about controlled access versus open. Open access doesn't. Controlled access can still be open.
I think the consideration about DCP, we need to consider two other questions. One is what do we mean by data? So there are many different types of data. So that's one thing, not totally including metadata. And the other is because currently there are multiple funding sources supporting HCA-related work. So some funding agencies have their special requirements or restrictions on what kind of data, how they can be released. So ideally, if there's a unified platform, that would be fine. But practically, probably there should be other options. I suggest.
I mean, I think in response to that, I'd say I think there's a good cohort of people in this room who would start to be able to work through some of these requirements. This is the sort of information that the DCP is really keen to hear about, things that potentially put barriers in the way of sharing data with the DCP. So the representatives in the audience here, as well as wider, would be really, really keen to hear this. You know, I think if people have got issues that they would like to raise here, we'll do our best to answer them, I guess.
Shyam?
I think that's a really important conversation about controlled access versus open access and so on. And we are in touch with Zina also about this, and I think some of us sent her emails about what kind of restrictions we have on open data sharing. And so, yeah, I think, as Al mentioned, it would be good to come to some conclusion on this in the HCA, but I don't think we can resolve that today. But I would encourage everyone who wants to contribute to the HCA DCP to talk to the DCP reps over here and communicate what legal restrictions you have in your program in your country on really open data release, and that'll really help them formulate the policies that'll work for everyone. But if we don't communicate our requirements to them, we don't communicate our constraints to them, they can't sensibly formulate policies that'll work across the world.
That's absolutely right. I would say legal, like national regulations, as well as just funding agencies as well. Different funding agencies have different stipulations. So we'd be really keen to hear about them. The more information that we can assimilate, the easier it will be for us to find a solution.
And I really want to thank the DCP members for making the trip here. Very much appreciated.
Still quite tired.
So we get a new regulation laws in China about the human genetic resources. So currently it's still not clear if the single cell data is under the regulation for human genetic resources. At least from my understanding, we probably have to under regulation on that, which means all the human related data, including the single cell sequencing data, has to get a special approval process before sharing outside of China. So this is one of the very important reasons for the Great Bay areas that the data at beginning, before get approval, we can sharing inside of the local consortiums instead submit directly to the global databases. So this is probably a common problem for the countries like China have such kind of regulation in human genetic resources.
Can
Go ahead.
say one more? So one way maybe to address this issue is our lab's having discussions with DCP because we have a mature data portal as well, and we're just simply leveraging the effort that DCP has put in terms of pipeline and so on, but then we can host the data effectively through our website as well. So other parties can have a similar model where we don't duplicate the effort, but you can still get away from all these global issues of restrictions and so on, right?
So your portal is presenting data from that's already in the DCP alongside
We
your
haven't
own
got that
data.
far yet. Yeah, but that's the idea.
Okay. Okay.
I just wanted to ask a clarifying question about the new regulations in China. Is it that the data can't leave the cloud region of China, or is that it can't be shared with anybody outside of China?
It can be only. They actually have a approval process. If you publish the data, the paper, finally you can able to update the data outside of China to be sharing globally. But before that approval, the data are not allowed to cross outside of China. So it has to be strictly limited only in China. The foreigners are not allowed to access to the data. The approval process currently is combined with the publication. If you have evidence that the paper is going to be accepted and they understand the concept, the information of the paper is not related to some sensitive things they are worried about, then they will give approval to release the data. So then that will be the later stage of your research. Then will be too late for sharing the data in the community. It's better to have an early one. That's why the local communities is better to have using some kind of data sharing platforms to working together. I think that's why I think the local consortiums, if they can have the similar structures as the current DCPs to adopt it locally, will help to the different local groups to sharing the data in the same kind of infrastructure.
Me myself has one point to make. Okay, okay. Small point. Okay, so usually when we talk about like sharing data, that within our Japanese consortium we talk about sharing two types of data. One type is like a personal information sensitive data. It's like really the fast QT alignment. So the second type is just a gene count matrix. Okay, so I assume that also the legislation, the regulations on protection of two types of data would be rather different. Okay, so that we are trying to make all those private, personal, identifiable data as controlled, as possible if that cannot be opened. And we also try to, on the other end, all the count matrices, if the collaborators themselves agree to open the data, and as well as in the ethics application, which is usually easier to open those data, then those will be 100% open. This is our approach. We separate the two layers very clearly.
So this is the current stage that we are not clear about the regulations, because from my understanding, if the data itself is not contain any like self-information or not able to convert to any personal information, this should be able to sharing. But at least from current communication with the China Ministry of Science and Technology, they believe that those data is also sensitive and should not be sharing. So this is still in the. They didn't have the detail explaining and the protocols of how that works. Currently, seems like it's a crazy things, for me at least. All the data is not out. Yeah.
Everything.
Yeah.
Hello. Oh, you are. Yeah, I've got an observation. I'm not quite sure whether I've misunderstood something or whether it is actually something real. In the data presented this morning, it said that there are order of magnitude about 10 million cells in the DCP. Is that right? I mean, it said there was, at least there were 10 million, there was a reference to numbers of cells that added up to around 10 million anyway.
Yeah, so that I think is that's the data that's available in the that's part of the Human Cell Atlas, not the DCP. The DCP is about 4.5 million cells at the minute.
So the reason I mention it only is because if you look at the number of people involved and the amount of money being spent in single-cell research, that's an incredibly underwhelming number. And the rate of the technology and the throughputs of the workflows mean that you could generate that, I don't know, in a few weeks. So I don't understand why in 3 years the number's so low. Is it because people have lots of failed experiments, or is it actually that there's a very high quality control threshold? Is it that people just don't want to give the data to the Human Cell Atlas? It's just a low number.
It is a low number, and I think I mean, so I should defer to some of my wrangling colleagues about the sort of their experiences like from the coalface. But I think the real answer is kind of a blend of the things that you're alluding to. But I'd be interested to. So we had one person in the audience who tried to submit data to the DCP. So I guess I would bounce the question back out to the floor and ask why have we just got one person here? Like what are sort of some of the blockers of people contributing data to the DCP?
I think at least in Australia, the collaborators, there's other Australians here. The ethics just don't allow, the consent hasn't been written in such a way that you can put in raw low-level sequence data. It would mean going back and getting a new consent. It would also mean getting it past the IRBs, and that's a real roadblock. Sharing count data is easy. It's trivial. Now, so if this is behind controlled access, then we've got no problems with that. And another solution is if you've got a DCP instance. So if you can run up an instance in China, for example, you can put through exactly the same pipeline and then share the count tables, assuming that eventually get it past the roadblock of sharing count tables. That might be a way of doing it, or having some way of a communication layer between them. But anyway, that's my point.
I should add, it's the same thing for us. The open consent is something we don't have for any of our existing data sets. So we have to start from scratch. We're going to apply for a new IRB approval, and new consent forms, and then the new data set can finally be submitted. I presume it's similar constraints for lots of people. I want to pick up on something one of you said. So there's so many cells in the DCP and then so many cells in the Human Cell Atlas. Now, very early on, I had the impression that the DCP was the Human Cell Atlas to the extent that, if there's a Human Cell Atlas data set, it's on the DCP and vice versa. But are there Human Cell Atlas data sets that are not on the DCP?
Yeah, so I think this feeds into the same thing, basically. So the ambition of the DCP is to have all of the data for the Human Cell Atlas. But I think we're seeing some of these challenges with actually with practically collecting that data. But yeah, I should open it up to my colleagues as well, if that's consistent with their observations.
So I think it just kind of goes back to that same thing, the consent. So I think there's a bit of a distinction with the Human Cell Atlas in association with papers, and the DCP was kind of being developed in tandem and a little bit after this came out. But I think consent's been the big thing that we've heard, is that complete openness has been a bit of a problem or a bit of a blocker for a lot of these. So we saw obviously a large number of cells in the Human Cell Atlas related to that, and then the distinction of the DCP outside of that. And I think also just trying to kind of build as there's new innovation and kind of on the pulse of the new science that's coming out.
And I think primarily we've been trying to get the healthy human primary data in. We do have a bit of mouse data, but we don't come across, I guess, a lot of primary healthy human data. So that's also maybe why there's only 4.5 million.
Yeah, I think a little bit of separation of the disease, and we're starting to kind of work towards that, because obviously a lot of people are focused on disease and research with around that.
Just what level of quality control do you guys apply to data that's coming into you, though? I mean, do you actually reject a lot?
Not really. Essentially, that's why we're usually at a lot of these conferences, is try to get people, now that we have a DCP ready to go, to essentially get them to come submit to us. And we're pretty much open. We're willing to work with people. So we're not rejecting really anything at this point. We may archive things if it's not applicable to what we have, or if we don't have pipelines for it. We generally are trying to not reject anything, and we may just put essentially something in an archive or an icebox to hold up until we get something ready for, or until essentially we're able to bring it in.
To clarify that distinction, if I have a data set and I want to be in the Human Cell Atlas, but it can't be in the DCP, how did that happen? I guess there are some data sets in that category, right? How did that happen, and how can my data set get there if it can't go to the DCP?
When you say it can't go to the DCP, are you saying that you have like consent issues, or is
consent
okay.
issue.
So at the moment, again, this is something where we're trying to come up with a sum of all the different consent issues from around the world, where there are things like my data cannot cross our country's border, my data has to be rejected at any moment when I say so. I'm having trouble with IRB consensus. There's a lot of different issues with consent, so we're trying to decide at what level do we implement, and this is an ongoing conversation that we've recently started. But there are other issues where people generate data for the Human Cell Atlas that is very interesting, but maybe it's not biologically relevant because it's a model system that was done to try and figure out normalization, trying to reduce batch effects, variation, things like that. So there are other reasons your data is for the HCA in the long run, but maybe not biologically relevant for building an atlas. And so maybe it doesn't belong in the DCP in that case.
So then all the HCA non-DCP data are non-human? Or are there any human HCA non-DCP data?
No. I think there's like a human melanoma data set, and it's disease, so that's part of the HCA, but it's not part of the DCP quite yet. I think the only data that I'm aware of that's not in the DCP is disease data, and I might be wrong.
We do have some, but it usually has a human component. I also think it's this idea of an atlas and an archive, and trying to kind of have this distinction of, are we taking everything in, or is that used to build the atlas? And I think there's a bit of distinction with that, and that's something the larger scientific community we're trying to kind of figure out as the DCP.
Another thing is that in the beginning, we want to build basically a reference of healthy human tissue, and healthy human tissue where you do a survey doesn't necessarily get you that paper and grant funding that a lot of people want. So it's actually kind of hard. So we find a lot of perturbations, a lot of novel sequencing methods, a lot of disease tissue, things like that, that aren't necessarily ideal for building our first reference healthy human cell atlas. So when we are presented with an opportunity to, do we put this data set in or that data set in, we'll always pick the primary healthy human tissue when available. So that's another thing to consider.
I think I would just stress as well to go with this. I don't think there's there's not really a question of is there data that can't go in the DCP. There's a lot of issues to work through in terms of practically getting it into the DCP. So there's not a blocker here. It's just or there's no like hard cutoff that is like this is not going into the DCP. It's not good enough or it's not the right sort of data. But there is questions around prioritization and questions around issues to work through, like legal regulatory issues around consent. So we're not actively turning anything away, but some things take longer than others.
Certainly.
So I was wondering, since there's lots of data where we have consent issue because they were collected retrospectively, are there any discussion going on to change some kind of rules for those and maybe having a more stringent criteria for prospective samples? Because if someone has collected data with a different kind of IRB or consent form, it cannot be changed at this stage, right?
We do ask folks to start contacting the Wrangler team as soon as possible at the point of, even getting the donors, the samples, et cetera, so that we can start to work out a lot of these kinks moving forward and hopefully learn as we go on. We have run into things where, consent that we thought was good at the time of collection, it turned out later was not, and we've had to reject data and things like that. So basically a set of best practices of what do you need to donate data to the Human Cell Atlas does seem like a very reasonable thing to put forward.
I think many of our discussions are more on the strategy or policy side. Probably we should pay more attention on the technical side. I guess that would be more fruitful for this breakout session.
Yeah, sure. Sure.
But can I jump in and ask one more question?
Go ahead.
I just want to understand a little bit more. What are the metadata? Is there a minimum metadata that you guys put on it? And is it sufficient to be used later on? Because there are existing, TCGA and other consortium where there's beautiful omics data in there, but it wasn't sufficient metadata to make it useful.
So I will try to kind of answer that, and then also maybe do a summary if we want to move towards other things. But we do have metadata. We try really hard modeling them and work with different communities to make sure that makes sense for them. And so we're trying to anticipate use cases before that they happen. We have it online available, and I can personally come to you and make sure you have a contact so you can talk to them, because the way we work in a lot of ways is going to specialists and asking them questions and making sure it makes sense. But we do have a schema, and we do have things that are required and not required depending upon the barrier of entry. And I look forward to talking to you more about that. Also, I think it's important historically to remember that the DCP was originally started with the conception that it would be open source data, open access data in general. And I think we're learning more in the scientific consortia about that, right? And we're seeing that there is more to it than just that. And so what you see right now is the DCP in an honest effort to be here to hear about what are the kinds of things, what are the other settings, other kinds of data that we need to understand and see, and hear about those blockers so we can move forward with you. And so as you're having these kinds of conversations, just keep in mind that where we're at right now is not what we're going to be in the next year. And these kinds of conversations helps us get to the right place quickly. So I really appreciate that.
The goalposts have moved a lot over the last 2 or 3 years.
We had original intentions. They were great. And let's keep moving forward together to get to the next ones.
I'll say that into the mic. Yeah, so the goalposts have moved, not just on the Human Cell Atlas, but in genomic data sharing, genetic data sharing standards across the globe, right, over the last 2 or 3 years. It's been a huge change.
So if I can make a comment, a lot of this issue with ethics and data sharing is actually present in a lot of human data existing, right? So for example, in Australia, there's a 45ME up study, and the constraint surrounding that set of collection is is huge, right? It's certain computer, certain firewall, and I believe, I'm not an expert on this, there are actually computational data strategy to how to go in and out of this kind of control setting to actually extract useful information. And maybe we could look at what people are doing with those clinical data. And it looks like this is the same challenge we're facing at the moment.
Yeah, so we should. Do you want to move us on? Should we take this up later? Yeah.
Last
okay.
comment. Yeah, on this. Yeah, you could reply if you. Yeah. Yeah. Cool. Well, thank you. So DCP is important. Data deposition is important. Okay. So we could continue the discussion. So now you know who to talk to, right? If you have problems, right? So anyone who talk on here, the DCP experts. So now maybe we could move to some other topics, which is also very, very important, because we also have some developers that is actually created some very useful softwares, and potentially that if you haven't touched those software created by CCI, probably you should take a look at that. So one of them is Cell by Gene. So anyone actually used the Cell by Gene software? Except the developers, okay? Except the developers, okay? So, anyone? So, no, one, two, right? Right. Right. Yeah. So it's a different thing. So, right. So that why I brought up like Cell by Genes is because if we talk about like standardization at the end, if we want to deploy your TSNE map or UMAP on the internet, on the web-based platform for your collaborators, then what are you going to do, right? So you develop your own little server, or you use something that is developed like more standardized by the CCI platform or not. Okay, so that's something that I think worth discussion, especially for some of the local consortium, like within the Japanese consortium, for example, that we always have to deploy our data, that the collaborators provided the samples, and we have to get feedback, what is the most standardized and most meaningful way. So if any one of you has experiences on that, maybe you could share. Okay, so on deploying your single cell data to your collaborators within the consortium. You just send a JPEG of TSNE map for your consortium member, or what do you do, right? So, microphone, please.
So yeah, so we use Jupyter Notebook, and I think that's very helpful, but we have not done Cell by Gene.
Before Cell by Gene, I had my own solution. There was a JavaScript-based exploratory tool, but now I just give people Cell by Gene. It works pretty well.
It works pretty well.
Yeah. I recommend it.
In case if some of you haven't heard about Cell by Gene, so that it's very similar to the 10x Club browsing interface, but you could put it on the web, way more flexibility. You could select the cell clusters in a more flexible way. All right, wait, one of the...
I thought I'd also mention that there's a UC Santa Cruz cell browser.
Right.
That's come out of the UC Santa Cruz Genome Browser team, engineered by Max Hausler. And it is a way to take your data where you have your XY coordinates, et cetera. It can run through Seurat, ScanPy, different reduction algorithms, and allows you to interact with the cells as well. It works similar to Cell by Gene, but it produces a static HTML output, so you can have that, host that somewhere and reference it in a paper. You don't have to run a server or anything, it just works. So.
Excellent. May I ask, I haven't used the UC Cell browser, so does it has any genomic component in there? Say, for example, if I've done a TechSeq, for example, so the identity of the peak is super important for interpretation. So would the cell browser link back to some sort of genomic track that you could see the peak where it is?
At the moment, I don't know if that's integrated, but the developer would be very interested in hearing that sort of feedback. He's actively trying to make the tool suit people's needs. So if you bring that up with him, I know he'd be interested.
Excellent. So anyone has anything to share on this aspect, like sharing data with your collaborators? Go ahead.
Just food for thought for the group. The data coordination platform has ways of interacting with portals. So the data is in the system itself, and then you can take that and you can work with it in your own portal, for instance. I think it was mentioned a little before. It'd be really exciting to see how this group could, for instance, have their own branding or have their own experiences they give users with the data that's been processed by the DCP and then made available so that you can show it. That's a real possibility, and I think that would give a lot of personality and enable this community in a lot of ways on the science that you're trying to describe to people. So just keep that in mind as an option of something this community could do.
Excellent. Go on.
So I just like to share my own group experience about the Shiny apps that we share for biologists to use. Usually, if we share for 1 or 2 people, that's okay. But if we want to share for 10 people, then the server is too weak for 10 people to use at one time. So if the CGI can provide us the kind of server that can host the kind of web tools or interactive analysis tool, that would be great.
What do you mean? You mean CCI provided server for you to host your Shiny
to
app?
host the apps.
Good suggestion. All right. Okay. Data visualization and anything to share more? If not, we might talk about something also very important. It always hangs in our mind, just like a cell type annotation. Okay, so that I probably all of you would be aware there are like multiple different types of softwares on the market and their benchmarks, and whether that as part of the Asia HCA, like a community, that do the community need some sort of more official guideline for what is the best practices for say annotations? And this is something that I think it's worth discussion. Okay, so it's because that I'm sure everybody have their own very unique way of doing say annotation in combination of manual curation and semi-automatic unsupervised matching to some sort of databases. Okay, so if anyone of you has experiences across your own consortium that on standardizing and cell-type annotation, I think it's worth sharing. Alistair also made the annotation software, so.
I think this is a very important question. I do not generate data by myself, but I really look at many published data. So it's very common that different papers use different terms in the annotation, and the terms at different resolutions, and also so there is a urgent need for building up a kind of a controlled vocabulary for the whole thing.
I know that at the Tokyo meeting, Peter Karchenko was talking about this annotation pipeline. I think it was associated with the DCP. Now, with a standardized sort of annotation. I was wondering if there's room for some sort of dream-like challenge, because there's multiple groups in this audience who have developed their own annotation tools. You'd have to generate the data. You have to generate a new unseen data set to actually run it on. But in terms of pushing things forward, everyone could collaborate. Everyone could be a co-author on a paper where we've all tried these different methods, and these are the methods that perform better. In terms of the controlled vocabularies, we've been using cell ontologies because we use them in Phantom 5. They're not perfect. These are starting points. They need to evolve as you get finer and finer grained definition of the cell lineages. But you definitely need some form of structured ontology to capture it properly. That's my opinion. Sham.
I think having a competition is extremely useful for something like this because there's so many methods, like you said. One thing we've struggled with when we're benchmarking our own algorithm and other algorithms is where's that data set, where's that golden data set that we can use as a benchmark? And really for single cell annotation, it's tough, right? Because when you profile the cell, you destroy it. And so how do you know what its identity is and to generate the ground truth? One could arguably say, well, just use CiteSeek and the protein antibody tags will tell you what the ground truth is. But we've looked at some published CiteSeek data sets. It's fuzzy. You analyze the CiteSeek data set this way, you get this ground truth. Analyze it that way, you get a different ground truth. If you change your QC cutoffs, you get a different ground truth. There's ambiguous cells when you look at the CiteSeek data. You don't know what cell type that is from the CiteSeek data, right? So really curious to know what people think is I think it's really important for the community to have such a ground truth. What would it look like? How would we generate it?
Could you potentially do it with like lineage traced mice or something like that, where you know what lineage they came from? I don't know. You'd have to do it. It'd have to be multiplexed lineage tracing to do it properly. Yeah, I don't know. But I still think the main thing is we've got all these different competing methods, and I think having some benchmarking is critical. If we can be smart and figure out what this ground truth really should be, then it would be really cool.
Yeah, I think like maybe like currently the more important thing is like practically within certain sizable consortiums that is there anything that they've been doing for like practically usable within, say, 10, 20 collaborators that everybody would be agree upon? So that might be something that some other consortium could borrow the experience and sort of share what is the best practice. So we ourselves in the Japanese consortium, we haven't, came across at that point yet. Okay, so large scale annotation of many cell types, but so far our experience is dealing with individual collaborators, tissue per tissue, and we still require a lot of domain expertise for that particular tissue to come up with something they think is reasonable. Okay, so this is so far what we've been trying to do. Okay, so but is there anyone who's done something in more larger scale on a larger consortium that probably would work to share the experience?
So
I think it's an unsolved problem. Sorry.
Yeah, sorry. I think I was just going to say that I think it's a really hard question. I'm a statistician myself, so I played with a lot of these kind of tools and actually benchmarked a lot of, but the challenge is how do you actually create that benchmark data set? The real challenge is there's still, if you create such data set, what is to say there's unknown or novel cell type that you haven't discovered, right? Yeah. And that's going to be where the money is, right? The T cell and the B cells, they're always the easy one. It's that small population where it's going to make the difference, refining that very small notation. And that's where I think it's going to be hard to look at comparison or benchmarking. Maybe it's doable. I think it's worth brainstorming and see how, as a community, we can come up with something like that.
Sham?
I have one suggestion. I want to, if you just mix a whole number of, using MuxSeq, mix a large number of cell lines with characteristic mutations and into one single pool, and then use demuxlet to say which cell line this thing came from, and the cell lines are similar enough, and some are rare, some are common, so on. I think that would be a nice benchmark data set.
Are you talking cancer cell lines with messed up genomes? Because they cluster separately really well.
They don't have to be cancer cell lines, right? So maybe cancer cell lines, some of them are too easy. There are some cancer cell line data sets already, and same cancer type, different cell lines, it's actually too easy to separate them. So they're not a great benchmark. But you could have cell lines that are closer to each other, or I think if we have like 30 cancer cell lines or 20 cancer cell lines, they'll start to look, some of them will start to look like each other, or non-cancerous cell lines. They will still be genetically different because they come from different individuals. And so DMUXLET can separate them. And then maybe that'll be a more subtle and more challenging and interesting benchmark. And then you mix the cells in different proportions, so some are rare, some are common. And at least you know that your ground truth is real. And it's unambiguous. And then we can see what the algorithms can do on that.
So because we've been running a data portal for a long time, so for the samples which have been annotated through surface markers and so on, we just have normal tables, SQL tables effectively. But for single cell data, we can't quite do that. So one idea we're toying around with is having a basically a computed or predicted cell type definition. So that's sort of more flexible, and you might have a score even against that. And the score might reflect how certain you are that you're predicting it to be that. And there might be some metadata associated with the prediction that you've made and what kind of method you used for that. So you'll need a completely sort of different data structure behind the scenes, but idea is to present the user with as much information as possible in a kind of more summarized manner, so that they can make their own decisions about it in a way.
So do you mean having a more trackable annotation, like a metadata, so why that particular cell is being annotated in certain things? Is that what you mean?
So we're talking about computationally annotating something through clustering or whatever predictions you're making, and making that more transparent to the user and saying that we predicted it this way because of these methods.
More trackable. Right. All right. So, anybody on cell annotation? So, if not, we could jump to another topic. Okay. So, and other than data sharing, cell annotations, and visualization, and one probable discussion topic would be that you can imagine at the end in a human cell atlas that you're dealing with potentially the millions and millions of cells, that how could that unit to be represented in something that is more interpretable. Say, for example, that we've been talking about something like, a metacell or a pseudo bulk cell type to be represented in certain reduced dimension reduction, right? So, and the simple idea is it's impossible to display, 10 million dots on your screen, right? So that probably some of the consortium already have some of these experiences if you're generating, large atlases, that in general, how to these problems to be dealt with. Okay, so if any one of you has these experiences, probably you could share with us.
Just about the 10 million data points. So Saskia Freytag, she's developed this hex binning method to reduce the dimensionality. It simplifies it. Yeah, so some of these, potential solution to that problem, right? So there's anyone that has experiences in that, instead of, going through, the data points at every single cell, that how about grouping them as a meta cell for representation? So that is something that I'm expecting even at the end of the human cell atlas, what is being, to be like with a manageable number of unit. Okay, so it's not just about, so you have cell type, you have cell state, but those are more biologically defined. Okay, so but could those cells be grouped as some other concepts, like as a meta cell, which some of the people already proposed that way of doing that? Does any one of you here has any opinions or experience on that
or thoughts on that. Okay, so potentially that we could be discussed here is because you also think about, like, for example, the cell by gene, for example, right? So the input data is being input as every single individual cells, okay, so display on the t-SNE map, okay, so, but could that be more dynamic, or calculated on the fly, that you could group the cells together and sort of reduce the dimension to make the data more manageable? So that anyone have any opinions on that? Or experiences?
I've noticed that for a lot of in establishing new biotechnology, we're focusing on cell. But when I actually work with my collaborators, most of them actually ask about cell type composition. That's their real interest, is what are the cell type in the individual and what is the composition? So is it 20% T cell, 40% T cell, and what is the variability associated with that composition? So to me, eventually, that's what people want to know, is the composition and whether the composition can predict blah, blah, blah downstream about an individual. So is it the cells going to be the unit or the individual at the end is going to be the unit? So I argue that some work, and we do actually have some work on differential composition, is really where this thing is the next step going to.
Yeah, I think that's a very separate question. So it's cellular composition, in my opinion, it's about reproducibility, it's about experimental biases that Alistair already showed, right? So the cellular composition is just a reflection of how you do your experiment. Okay, so, or, it has certain randomness. When you process the sample, some of the cells will be lost. And maybe Alistair has a better answer on this.
I agree with Jean. I mean, so with the kidney experiment, we were able to do two types of analysis. One was differential expression within a cell type, and one was differential population analysis. Differential population analysis is really hard to do unless you have true replicates and you know the sampling bias. So if you're taking a tumour, depending on which part of the tumour you're sampling from, you may get lots of T cells or lots of necrotic cells. So until you have an estimate of the variability within that sampling, and then you look at the variability across patients, then you can start to look at how many patients do I have to look at with this level of variability to reliably say that there is a twofold difference in the number of T cells of this flavour. And to be honest, I think you're going to need massive numbers to do this properly. Yeah, it's the sort of the holy grail kind of question. It's like, okay, tell us what the composition of this thing is, and it's going to tell us if there's a cell type that's diagnostic or not. But you need lots and lots and lots of data to do it properly.
All right. So we were, so before we talk about composition, right? So we were talking about this, in terms of making an atlas, what is the, ultimate deliverable, okay, in your mind, right? So that, and probably is that we are reaching a stage that we could annotate a cell with certain state, with certain, cell type. Okay, so but what we expect, this is not the end of the story. This is the beginning of the story, right? So, and we think about it within our Japanese consortium a lot, that if you have something like, for example, like a tubular mirrors, okay, so, and what they deliver is essentially a big table. Okay, so per cell, you have some annotations in there. Okay, so from a user perspective, also from a bioinformatician perspective, okay, so how much can we do about this level, right? So more than just per cell, we have some identity, some spatial information, but how could these cells, to be represented in a more, interpretable way, that some of these ideas could be feeding back to the software engineering team in here, that potentially those concepts could be fit into the cell by gene software. Okay, so does anyone has any ideas on that? That probably we could discuss about it. Take the mic.
So we can consider a cells as an individual in a big community. We can see the cells, they are talking to each other, communicating with each other, so that we can borrow the idea of Facebook, so to see how the cells, they interact with each other and form this community network. And then we can overlay this information on the cell type, and then to see which cell type is the key player in this network. What other cells it interact with also defines its cell type.
Yeah, so some of the concept is currently lacking in the software making process actually. So can we add edges between the cells?
Yeah, add the badges. Yeah, add the badges to the cells.
So
Yes.
because like most of the software displaying like a TSNE or displaying a UMAP.
Yes.
So usually that the edges between the cells is ignored. But how you build the edges is another question.
Yeah. So usually people use these ligand receptor interactomes to build the edges to connect the cells. Because that is the main type of cell-to-cell interaction, which is by the ligand receptor bindings. I believe that there might be other types of cell-to-cell interactions. Yes, visualization.
So actually there are kind of cross-mapping of the cells, trying to find who is closer to which cell is closer to which cell. So there are some methodologies for this, but how to visualize them and how to make sense of them is a challenge.
Yeah. So I just believe that it's like a person who we interact with, or like who our family or friends are, also defines who we are. So maybe this also applies to the cell level.
Get your idea. So it's like a cell type annotation taking into the account of the interaction with other cells. So it's just more complicated. Okay, so so probably we're running out of time. That we might have to close the session here so that we could prepare some slides for reporting. So thank you very much for joining the breakout session.